Bhuvshat, madam?
Yes, ma'am.
Yeah, we can start.
Am I audible to all?
Yes, ma'am. Yes, ma'am. You're audible.
Okay. Good morning, dear delegates. So once again, we are back for our day 5 session of One Integrity Development Student Training Program organized by Yashwantrao Bhosle College of Pharmacy. So today we have with us eminent speaker, Mr. Ganesh Vagwe, sir. Sir, I welcome you for the today's session. Sir will be addressing us on session for the topic today is AI tools for drug modeling, making NCE design easy. With this, now I would like to request Mrs. Gayatri Atlekar, ma'am, to please take over the session and give us the introduction of our today's guest.
Thank you, madam. Uh, it's my immense pleasure to introduce Mr. Vaghwe, sir. Uh, just let me share his CV. Is it visible?
Yes, yes, ma'am.
Sir, Mr. Vaghuri, sir, is engaged in the diverse project leveraging AI and ML access across drug discovery, drug design, and regulatory affairs. He has developed many innovative tools integrating cutting edge technologies to predict bioactivity, classifications of regulatory variations, and optimize drug compatibility, significantly advancing research efficacy and regulatory compliance in the pharmaceutical sector. Sir is a founder of Sequient Innovations and Jane Force, with which we are going to be introduced in this today's session. He is also expertise in AI and ML applications in drug discovery and design with a strong foundation in bioinformatics and Python. He has also developed innovative open source tools for bioactivity predictions and drug compatibility, which is enhancing the pharmaceutical research. He has demonstrated ability to apply computational method for drug designing, significantly advancing research and development processes. He is also skilled in regulatory compliance, employing advanced technology to streamline drug discovery and regulatory affairs. He has pioneer or leader in a project management with collaborative approach in problem solving and innovation. He is effective communicator, capable of driving technological advancement through strategic collaborations. He has worked currently working with so many collaborative projects with different pharmaceutical industries and regulatory industries. He is expertise in AI ML, which includes drug design and discovery, also bioinformatics and computational biology, machine learning and data analytics. So with this core, so many core competences regarding AI and machine learning, deep learning, Python, even Viva Vault, scientific research, academic excellence, training and mentoring. Sir is also expert in molecular biology sector and algorithm development, again data mining and analysis, and along with that conflict resolution and analytical abilities. So he has academically qualified as a MPharm in pharmaceutics from Savitribai Phule University, and he has filed different patent applications for invention entitled pharmaceutical composition comprising tioconazole and terbinafine and method for the treatment of onychomycosis. So he has different derivations of patent in his name, in his credit. Also, he has number of open source project going on, namely GenForce, Drug Compact, Reg Classify X, and Neuromo Predict. So with this brief background, I would like to request Mr. Ganesh Waghure sir to commence with the session. Now, platform is yours, sir. Thank you.
Thank you, Gayatri ma'am, for this wonderful introduction. And this is my immense pleasure to be here and giving me this honor to, to present in this workshop on the AI and ML in the drug design. So thank you so much. So we can get started for the today's session. Let me share my screen. Does my screen is visible to everyone?
Yes, sir.
Yes, sir.
Yeah, okay. So let me open the presentation mode. Okay. Uh, Basically, in the today's session, we moreover focus on the actual demonstration on the certain software that we are going to be used for the drug design and other molecular docking applications, right? Before to proceed with the actual demonstration on the this particular softwares, let me we'll go through some theoretical aspects of this particular application, and once this theoretical aspect you are able to understand, then we are going to be proceed with the actual demonstration on all the software that we have been developed, or the other software tools that are available for the to use for the molecular docking and other drug design and the drug discovery approaches. Okay, so in today's session, we are going to be cover the basic introduction for the molecular docking. What are the different, tools that are available to perform the molecular docking simulations? The selection preparations of protein and ligand, and then we are going to be cover the demonstration on the Gendoc suite. Also, there are the other software that is the ADME prediction, toxicity predictions, bioactivity predictions. We are also going to see how to perform the interaction of the drug and co-former for the different type of solubility enhancement. So let us get started. So there are the different approaches for the drug discovery. Mainly that if you see, the first will be the fragment-based drug design, where you are having a 3D structure of the protein, but there is no ligand for that particular 3D protein. Then you can go for the de novo drug design and this scaffold hopping. We are also going to cover in this particular session, where we are having one of the tool that is the GenMol, which will generate the novel molecules based upon the selected targets and the train on the certain database. So this is called an fragment based drug design. Then the structure based drug design, where you are having a 3D structure and both the ligands for that particular receptor. Then you can go for the molecular docking and the dynamic simulations, which is called the structure based drug design. Then third type, that will be the ligand based drug design, where there is no 3D protein structures available for the for ligands. You are having a ligand, then you can go for the 3D pharmacophore similarity, 3D QSR, and the other thing that you can perform to select the best proteins for that particular ligand. That is the ligand based drug design approach. And the combinatorial drug design, that is the last one, where you do not have the 3D protein structure and there is no ligand which is available. Then you can go for the COM-BCAM high throughput screening and the virtual screening that you can perform for the selection. So this is the key area and the different approaches which is mainly used in the drug discovery whenever you are going to proceed for the drug design. All right. So let us understand what is the molecular docking. So molecular docking are nothing but the to determine the optical binding structure of a ligand, or we can say that is the drug candidate, or it is also a small molecule to a receptor. That is your receptor. It will be a target or the protein or the DNA. We have to quantify the strength of that particular ligand and receptor interaction that has been formed. So for to quantify this type of ligand and receptor interaction, there are the different type of the scoring function that is available, right? So we are going to determine the binding energies, which is an delta G that we are going to be determined, right? That is the Gibbs second law, right? So we are going to be determine those are the binding energy that has been formed once you have been performed the different types of the molecular dockings, right? So if you see there are the very basic molecular docking understanding, that if you see the lock and key model, right? We have to see how your ligand will going to be fit in that particular target or the receptor, or in the cavities or the different type of the cavities we can say available in the protein. So your ligand will going to be bind in that particular cavities, and that will form the complex. So that is the similar type of the lock and key model over here. So after this particular complex has been formed, so the different scoring functions are there to quantify the interaction between this protein and ligand that has been formed over here. So we will see in detail about the what are the scoring function and the different type of the scoring function that is available for the different software that we are going to use in the molecular docking application. So let us understand this particular thing in more detail. Like there are the different principles of the molecular docking. As I said, first there are the lock and key model, where in the receptor you are having the fixed shape of the cavities and the pocket that is available, where your ligand will going to be fixed in that particular cavities, and that will form the receptor ligand complex. So this is a simple lock and key model to understand the better molecular docking simulation. Then there is a next model, that is the induced speed model, where your ligand will bind to that particular receptor in incorrect conformation, but that ligand will what will do, that will make that particular receptor to adjust its shape, and it will form the stable conformation like this particular way. When the ligand has been first bind to that particular receptor, so there are the different the amino acid in the protein that will adjust their shape and form the stable conformation, that is called the induced speed molecular docking. The third approach is that is the conformational selection model. In that particular, we are moreover use this particular model in the when you are using the different type of open source software, that is the molecular Autodoc Vina or the Autodoc, right? Where you will generate the different type of ligand conformation. So ligand so the base conformation which is fit in that particular receptor cavity, that will be selected over here. So ligand chooses the best conformation and bind to that particular receptor cavities according to the different cavities and the pockets that are available in the protein itself. So this is the conformational model for to perform the molecular docking simulation. There are also the different concept, that is the rigid docking, where your both the receptor and ligand are the rigid, they are not the flexible. So in the defined shape they are going to be docked in that particular receptor, right? Then the flexible rigid docking, where your ligand is flexible, it will generate the different conformation to fit in that particular pocket or the cavities which is available in that particular receptor. But in that particular, your receptor will in the rigid state, only the ligand will be flexible, that will generate the different types of the conformation and fit into the pocket or the cavities that are available into the receptor. Then the flexible docking, where in that particular case it will your both the ligand and the receptor both are the flexible state. So in flexibly they are going to be dock with each other, and that will form the stable conformations in the after performing the docking. So this is the basic understanding, like even in the body, in the physiological condition that we are going to be say, this is not an the fixed or the rigid step. Every condition that is continuously simulated, right? That are not the fixed or the rigid conditions. So these are the various factors that you have to understand while you are going to be performing the molecular docking simulation. Based upon your study, you need to select the model or the which type of molecular docking you are going to be perform. So next is the molecular recognition. So this is a very basic concept that you have to understand. Like every cellular process is happen due to there are the interaction between the molecules. So when you see the physiological condition where every cellular processes, whether the activity or the any condition that are going to be formed. This is because of there are some interactions where formed between the different molecules or the different groups in the body, right? So this all the interaction were going by the different intermolecular force of attractions. So there are the different interactions that were going to form, and this is all because of there are the different intermolecular force of attraction. So our goal in the docking that we are having an given structure of the two molecule, and we have to understand where this both the ligand and bond ligand and the receptor, whether they are going to be interact favorably. If they are favorably interact with each other, then we have to understand what is the orientation of this particular the interactions, right? Then also we have to understand what orientation that we have to give in that particular cases, which is maximizes the interaction and minimizes the energy. So that isn't very the basic understanding, and that is the main the objective behind to perform the molecular docking. And we have to see how we can maximize the interactions and minimize the energy, so that will be an more fit complex that will be formed during the molecular docking simulations. So these are the basic understanding. Then we have to the very important why there is a need of the molecular docking, and what is the basic significance, and what are the different key aspects that you have to understand while performing the molecular docking simulation. So first you have to understand what is the catalytic functions of that particular receptor, right? If that is an enzyme, so how the catalytic reaction that has been placed in that particular receptor. So did you have to understand. Then secondly, how to understand what is the regulatory mechanism of that particular receptor, whether that proteins were show the activity by joint by when your ligand will bind to the active site, or there are the different allosteric site which is present in the receptor. So you have to understand the regulatory mechanism of that particular receptor over here. Then you have to understand the cleft detection. That is a very important aspect over here. You have to understand what is the different pockets that are available, or the cavities that are available in your receptor. Then the geometric analysis, that is the shape of the proteins where your ligand will going to be bind. The available pockets, that is a very important, where your ligand will going to be bind, and it will form the interaction with the defined amino acids. Then the flexibility of your receptor, you have to understand whether your proteins are flexible or not, like what are the key areas in the protein uh, to perform the molecular docking. Then the very important, that is the conservations, what are the different amino acids that are present in the receptor, those are the conserve. I mean, like the amino acid which are the same, which is present in the different types of the other species as well. So this type of amino acid, when you are going to be understand, so you are going to be select the base active site to perform the molecular docking application based upon the selected or the conserved amino acid in that particular mm, uh, receptor. Then the surface characteristics, so this is also one of the very important factor. You have to understand what is the surface charges is for present on that particular receptors, because when we are talking about the interaction, so there are the different surface charges that are present on the amino acids and the ligands. So they are formed that to consider this surface characteristics is very important to perform the molecular docking. Then whether the negative charges or the positive charges or the different surface characteristics that you have to understand before to proceed with the molecular docking. Then you have to understand what is the interaction energy between the ligand and receptor. So these are the very the basic thing that you have to understand whenever you are going to perform the molecular docking simulation. You have to you have to understand this particular aspects of the receptor or your protein, and before proceed with the molecular docking, you have to understand this particular aspects of the molecular docking. Then you will also have this particular question that you may have to understand, like where will the ligand bind. You have to understand when, in which ligand, in which pocket or the cavities where your ligand will going to be bind. Then how will it bind? Then how strong the binding affinity is it? Then what is the role of the solvation and desolvation? Then you have to understand what make a ligands bind to receptor better than others. So that is also the key aspect. Docking is isn't about to only perform the simulation and generate the docking score, right? Your on only docking score, it is not defined whether you will get the better by activity or not. There are the other aspect of that particular molecules or the docking simulation that you will have to understand before going to proceed with the molecular docking. Then the translations and the rotations of that particular ligand. You have to understand the torsion, the selectivity and sensitivity, also the multi and off target. So these are the basic things that you have to understand before to proceed with the molecular docking applications, isn't it? So the next is the where the dockings go, right? So you have to understand. So this is one of the example of where the dockings has been performed. If you see in this particular image, if you see there are the some green bonds were formed. So these are uh, the frequently observed uh, interactions which is observed in the database of the CSD. Then the yellow color, if you see, these are the unusual interactions, but that is also observed in some of the CSD databases. Then also the red color, if you see, that is very rarely observed in the CSD databases. And if you see the detailed images of this particular interaction, there are the green type of interaction that is formed, that is the good for the affinity, and that can show the better bioactivity of the molecule. But if you see in the red color, that is the bad for the affinity. So you will have to also understand why this is then bad for the affinity. What are the functional group that are present in this particular condition? If you change the functional group in this particular position, then does it increase the binding affinity and for that particular ligand or not? So these are the basic understanding that you need to understand after performing the molecular docking application. Not only depends upon the screening based upon the your docking score, right? So these are the basic thing that you have to understand. Now these are the some of the aspect that we are going to be covered with respect to the demonstration that we are going to be giving for today's session. That is the y active site detection. Then because the identifying the correct binding pocket is then very critical and the accurate docking predictions of the drug design success, right? So the selection of active site, it is a very crucial step that you need to be select then very the important active site within the receptor and the pocket where when your ligand of choice will going to be bind in that particular position, it will show the bioactivity similarly to the when you have been taken that particular drug, right? So there are the different approaches for the selection of the active site. The first, that is the co-crystallized ligand based method. So this approach will understand what is the reference compound or the standard compound that is already there in for that particular receptor, and in the position where that particular ligand or the co-crystallized ligand, that is the nothing but the standard or the reference compound that were available in that particular pocket. So based upon that co-crystallized ligand in the X-ray structure of that particular protein, the same active site will be selected for the perform the molecular docking applications, right? So this is one of the method for selection of the active site based upon the co-crystallized ligand based method. The second one, that is the cavity and pocket detection algorithm. So if you see there are the different uh, web servers available which will detect the different cavities that is available within the that particular receptor where our ligand of choice will going to be bind in that particular pocket. So this software identifies the geometric pockets which are available in the receptor. These are the some of the example that has been mentioned over here. That is the cast P, then the site map and dog site scorer. That is these are the three available web servers to detect the cavity and pocket within the receptor. There are some moreover available uh, web servers, but these are the commonly used web servers for detection of the active set within the your protein. This also analyzes the surface topology and the volume characteristics of your receptor, and they will understand what is the different, the pockets or the cavities that are free spaces within the protein that are available where your ligand will going to be bind. So based upon that, they will give you the different pockets coordinates. On the basis of that, you can select those particular pockets or the coordinates. Yes, hello, hello.
Sir, it was by mistakenly unmuted.
Okay,
You can
okay, okay.
continue, please.
Yes, yes. All right. Okay. So the third approach that you can go with, that is the blind docking approaches. So this in this particular condition, user can select the entire proteins as in protein surfaces without any bias, where you can select as a whole protein as an binding site. That is the blind docking approach, and you can perform the molecular docking simulations for the entire protein. So that particular algorithm will by itself detect the different available pockets within the that particular receptor, and that will dock the ligand of choice and generate the different conformations for the docking simulation. Then the last method for the active site detection, that is the sequence and structure homology mapping that we can say over here, right? So this will be identified the site based upon the conserved residues across the protein family. So as I said in the last slide, that is the conservations, right? This is a very important. So we are having a different species like the humans and the animals and others, right? You have to understand what is the residue that is the commonly available in this particular across this particular species. So these are nothing but the conserved residues. Is if the same type of the residues that are available within the protein families, then you can select those particular amino acids as an identify those particular amino acids, and based upon those amino acids you can select the active site for the to perform the molecular docking simulations. So so that's why this is called a sequence and structure homology mapping. So for that there are the different type of options that are available in the software, like the constraint docking, where you can define the amino acids, and based upon the selected amino acid, the same the grid we can say for the molecular docking, the same grid has been generated to perform the molecular docking. So these are the all the four different types or the approaches that we generally use to perform the molecular docking simulations. So these are the various tools. The first tool that have been mentioned over here, that is the GenDoc suite, that is the specifically developed by the GenForce Technology Private Limited, which also uses the Autodoc VNA and Autodoc in the backend, but we have been simplified the so many processes and the different type of scoring function that you have been available within this particular software. And in the one click, in very user friendly, you can perform the molecular docking simulations, and you can also analyze the result. So we'll going to be see the actual demonstration on this particular software. Then there are also separately, user can also perform the Autodoc VNA, which generally requires the command prompting, and there are so many convergence methods which is required. Like you need to use the open bubble if your molecules are in the smiley and the mole file, that you need to prepare your ligand there are the so many conversion that then you need to be energy minimization, so that there are the so many convergence method that is required. The Pyrex is also an GUI based application which is also used as the same Autodoc and Autodoc Vina in the backend. Then the Open Bubble which is required for the conversion, the MGL tools, so this is a part of the Autodoc for the preparation and to perform the molecular docking simulation. Bioad Discovery Studio, which is generally used for the visualizations of your protein ligand complex, and you can also use for the protein and the ligand preparation as well. Then the leak plot, this is basically used to generate the 2D interaction images of the uh, a protein ligand complex, right? So these are the basic tools that you have to use across to perform the molecular docking simulations. So these are the three functions which is currently offered by the GenDoc suite to perform the molecular docking. So you are having a choice for the user. You can select which scoring function that you can select. So every scoring function having the different formulas based upon that that will score and perform the molecular docking simulations, right? The Vina, Vinardo, and Autodoc 4, that is the AD4 that we can call. So these are the different uses, the different types of the force field, and the different types of the calculation to calculate the binding affinity for the protein and ligand complex. We'll not go in detail for here because we have to, in the time constraint, we will also have to cover the actual demonstration on the software. Then there are the different steps for the molecular docking in our current software. That is the, you can upload the protein, prepare the receptor, you can add the ligand, and then the ligand will be prepared. Then you can configure the grid and you can run the docking simulation. So in the 4 to 5 steps, you can perform very easily for the molecular docking simulations. So as I said, there are the different types of the force scoring function. That is the force field based functions. So generally the Autodoc 4 and doc or goal that will use the empirical scoring function. Then the knowledge based function, that is different types of the function. There are some popular docking softwares, the Autodoc, Vina, Autodoc 4 and Pyrex. These are the all the open sourcely available uh, tools that people generally use for the molecular docking application. Then the Glide, which is developed by the Schrodinger, then the Gold and the MOE. These are all the commercial tools, or you need and the academic or the commercial license for this to use this particular applications, right? So these are the basic. Then in the protein preparation, there are the different steps that you have to understand. We will not go in detail about the protein preparation. We'll going to be cover this particular point at the time of the demonstration directly. You only have to understand in the protein preparation, you need to remove the water molecules except the critical, which is you can keep the water molecules for certain in cases where the water will play a very crucial role to forming an interactions bridges through the water molecule. You can delete the co-crystallized ligand ions and the solvent from that particular receptor, and also you can remove the redundant chains if there are the duplicate chains that are available within the proteins. And these are the basic thing that you have to consider for the protein preparations. So all right, I can just okay. Okay. Um, is my screen is visible to everyone?
Yes, sir, it's
Yes.
visible.
You are able to see the software that I have been open, the GenDoc suite.
Yes, sir, it's visible.
Okay, okay, all right. So see, this is the interface of the GenDoc suite applications that are completely developed by the GenForce Technology Private Limited. So once you have been installed these particular tools, if you see, this is how it will be reflect in your the shortcut icon that will be reflect in your PC. And once you open this, this is how it will be look like, right? So if you see, there are the in the left side panel, there are the different type of the options that are available for to perform the molecular docking simulations. So let us start with the to understand this navigation to perform the molecular docking simulations, right? So in order to start with the molecular docking application, first you need to upload your proteins, right? So once you click on this particular upload protein button, so this type of the pop-up will be appeared in front of you, right? So over here you are having a two option. If you already downloaded that particular protein, then you can browse the local file. You can directly click on the browse button and you can select the ligand that sorry protein that you have been already downloaded from the protein database, right? If you are not downloaded, then we are also having integrated the same approaches in this particular software. You can just click on download pdb button, then you can it will show in another one uh, pop up over here. It will ask you to enter the pdb id of that particular protein that you wanted to retrieve. So for an demonstration purpose, I'm selecting one of the 1U54 receptor, right? So if the people they are having the laptop with them, we are also going to share the license for this particular software as well for the people those have been joining for the today's session. They only have to request the license for this particular software, right? So once you enter the pdb id over here, then you can click on ok. So see that directly that particular protein has been retrieved from the rcsb side. So in this particular all the structure details of that particular proteins has been shown over here. The proteins contains the two chain A and chain B. So for latest example, like we needed only chain A where the active site of the proteins within the chain A that is available. In some cases, uh, sometime there is also duplicate chains are available within the same PDB ID. So for now, I need only chain A. So I am selected chain A over here. So there are the different preparation options over here that is in by default that is needed. The hydrogen atom that has been added, the water molecules that will be removed, and it is also remove the other hate atom that is present in within the protein molecules. And then you just click on the prepare protein button. Once you click on that, see the protein preparation has been successfully completed, and it will showing the message over here. You can click on OK, and the prepared protein has been you can see in the viewer itself, right? You're able to see the protein that has been prepared over here. And the same detail has been chain A, and the protein that has been prepared has been shown over here. The next steps, once you have been prepared your protein, that is the ligand structure, then you can click on ligand upload ligand button, then it will show in another one pop-up. So over here there are the different options over here given to upload your ligands, right? You can add that particular ligand from the you have been already prepared the mole file, or that has been saved in any folder. You can choose that particular. If you are performing, if you wanted to perform the multiple docking, so this is also one of the features that are available in the current software. You can directly select that particular folder if there are the 10, 20, 50, or the hundreds of molecules that are available in that particular folder that has been automatically retrieved. And if you are having in the smiley of that particular compound, then you can enter the smiley of that particular compound over here, and that will be down there. So for example, I will share one example. So I'm sharing one of the mole file that I already have saved. So see that particular molecules has been ligand has been selected over here. Now what I will do, now I will click on. So if you see, there are the different preparation options are there. The add hydrogen. So if you see, these are the mole for file format. In case of the smiley or the mole, that the ligand are in the 2D structure, right? You need to generate the 3D coordinates for that particular receptor. That means you need to convert 2D structure into the 3D structure. the ligand is also prepared like the CAMD or the skate that has some constraint energy. So you have to minimize the energy of that particular ligand. Also, there are the different types of the force fields that are available to perform the energy minimization for the ligand. So you are having a choice to select the which type of force field that you wanted to select to perform the energy minimization. So default there is an FF94. This particular force field has been selected, and there is also check for that is the check then fix the valence errors within the ligand itself. So once you click on this ligand prepare ligand button, so within the minute that the ligand has been prepared successfully, and you can just click on OK. So that is also the same will be if you are able to see in the viewer as well. The ligand has been prepared and uploaded over here. Now there are the different docking parameters like as I said, we are in current software we are offering Vina, Vinato, and AD4. So depending on the your applications or which scoring function that you want, you can select the one of the scoring function from here. There are the different exhaustiveness and the binding modes. So to perform the more better docking and you need to select the highest higher exhaustiveness, higher the exhaustiveness and the binding mode that will generate the more number of the different conformations for that particular ligand. So the highest the exhaustiveness and binding mode, it will always better to to generate the better posing and scoring for the molecular docking simulation. The other parameter that has been already determined, calculated depending upon the which the laptops or the computer that you are using, right? Then the next part that is very important, that is the configure grid box. So once you click on configure grid box from over here, or also you can click on this particular navbar option grid. So this is one of the pop-up that is uh, uh, appear over here. So if you see under the grid configurations method, we are having a different option, that is the automatic, that is in blind docking, we can say that cover the entire protein. Then we are having a based on the crystallized ligand, and the manual configuration where you can manually address the grid within the protein itself. So for now demonstration, I will select based on the crystallized ligand. So once you have been selected, it will ask me to enter the PDB ID. So I will going to enter the same PDB ID which the protein or the receptor that we have been taking. Then click on the fetch. So once you click on that, see it will it the same. It has been fetched the crystallized ligand which are available in that particular receptor. So over here there are the chain A and chain B. They both have been fetched, but as we have been deleted the chain B, so we only have to look into the this particular two receptors. So these are the crystallized ligand. You are also able to see in the small screen itself. So based upon this, you can select the which receptor or which crystallized ligand you wanted to select for the active site selection. Also, if you wanted to detail understand like what is the PTR and ACP, then you need to go to the main website, that is the RCSB site. Then you can open that particular mm, proteins over here. So let me open that particular protein. So this is the same receptor that we have been taken for the demonstrations purpose. So I'm just scrolling down to see. So these are the ACP, that is the phosphomethyl phosphonic acid, and adenylate esters. And there is another one that is the PTR. So you can see the structure of this particular crystallized ligand. So based upon the literatures and the this reference compound, you can select the which is the crystallized ligand for that particular receptor. So once you have been selected, so you need to click on select, then you can click on select, use the selected ligand. So once you have been click on that, then apply the grid. It will say the grid applied successfully. And in the viewer, if you see, so according to the selected crystallized ligand, uh, this grid will be calculated based upon the where the crystallized ligand has been already docked. So that is we consider as an active site for the molecular docking, because the same site, the the standard or the reference compound were docked in this particular position. So once it has been selected, you can see the details has been grid will be populated into the left side of the grid box configuration. So once it has been all selected, you can click on start docking button. So this will open the job progress buttons over here, and if you are able to see, it will show the overall detail progress for the that particular job. You can also close this particular, and if you see in the submitted, that is showing one job has been submitted. You can click on submitted jobs, that will show over here. See the if you're able to see those showing the completed the one ligands, right? That means the docking has been completed over here. If you did, you can click on this, you can see the completed the molecular docking over here. You can now you can close, and in the input we are having input section and one of the results section. So if you see those the docking that has been performed, those all the result were loaded over here. And if you see the last one is the highest, so -6.6 is the highest score that has been generated over here. And when you double click on that particular complex file, so if you can zoom out, you can zoom in on that. If you see over here, your ligand, the same ligand has been joined over here, and that has been docked in this particular protein and ligand complex based upon the selected scoring function and the protein and ligand that you have been uploaded. Now, the second thing we have to analyze and understand what is the interaction that has been formed with the protein and that particular ligand. So once you have been loaded that particular complex over here, the second thing that you have to do is that you have to go to the interactions, and you can click on show interactions. So once you selected that, if you see, you're able to see the interaction between the your ligand and the different residue of that particular protein. If you're not able to understand which is the ligand, then you go to the view. Under that the receptor color, you can change the receptor color. Now I've been changed the receptor color. You are able to see the name of the residues, then which type of what is the distance, and which are the different amino acid or the residues that has formed the interaction with the your receptor, and you can easily analyze this particular thing. You can also take the screenshot if you need it for the publication, or you can go to the file and save, click on the save image. So same, the 3D interaction image will be saved in your the specified locations, right? Then some of there are the other option is also, that is show and hide the labels. You can hide the labels that the residue name. You can also show the labels. So these are the different options are available. You can show the specific contact. If you only wanted to show the hydrogen bonds or the weak hydrogen bonds or the hydrophobic or the pi stacking, so you can select this particular option according to your choice, right? You can also show the ligand surfaces. So similarly, that particular ligand surfaces will be shown over here, right? And there is also one option, that is you how you as an publication point of view, that you needed the different the interactions tables, right? What type of interaction, which name of that particular amino acids, then the category and the type of interaction that has been formed, and the distance or the length of that particular bond. So under the interactions, there is an option for the interaction table. When you click on that, so see, this is the one table that will be open over here. And within this particular table, you are able to see the residue name, their distance, their category of that particular interactions, and what are the bond type of that particular interaction. You can just export this particular result into the CSV file, and you can save those particular result, and you can later on you can use it for the publication point of view. So these are the within the very simple one click, you can perform the molecular docking simulation, and you can easily analyze this particular result. Now there are the two aspects that you have to understand over here. The first thing, once you have been performed the result, after that you need to analyze this particular result. Then you can, there are the option for load a result option. You can just directly select the in the working directory, you can select the which are the complex that has been formed over here, and you can just click on that particular complex and open. So that particular complex will be loaded into the viewer, and you can easily click on and you can see the you can analyze those particular results. Second, in the bottom, if you see, there is an output directory over here. So when you install this particular software and activated the license for it, this will be an auto select this particular directory for where all the docking simulation that you have been performed were saved in this particular folder. So if I see, if I click on result button, so that will open directly that particular directory. And if you see over here, I have been performed this particular docking simulation. So for the each ligand, for the each type of ligand, it will create the separate folder for the docking applications. Like if I have been uploaded the A1 ligand, so the separate A1 folder has been created over here. And under this A1 folder, if you're able to see, these are the complex file that has been generated over here. In the single complex file, it will both that particular ligand, that particular pose and the receptor that has been present. You may use this particular complex file if you wanted to if you want and different different tools like bio discovery studio for the 2D and 3D interactions, right? Those you can use it for to analyze this particular result, right? So for now I can just open the same in bio discovery studio. In the one click you can open that particular same into this particular application in BioWare Discovery Studio. And if you see the same things you are able to see over here, you click on the ligand interaction, you are able to see the same interactions over here. And also if you wanted and show 2D diagram, you can just click on it, you are able to see that particular 2D interaction diagram. Now this is the choice. You may use this particular tool or you may use the leak plot. There are also other 2D interaction plot options that are available for the users, like the pose view and the pose edit. So these are the different web servers that is available to generate the 2D interaction diagram. Currently, the 2D interaction diagram has been not configured in this particular tool, but in the next version, definitely this options will be available for all the users to generate the 2D interaction plot for the same. Also, other option that is the view log over here, you can select or see the log that has been generated over here. So this is all respect to the JNDoc application that you can use it. Currently, you needed a license. We are providing, as we have been some discussion with the the staff, that we are also going to provide those students who are joined for the today's session. We will going to be provide them and the specified period of the license, pre-license for the to use this particular application. You just only have to request for the license on our email ID, that is the info@genfold.com. We will going to provide the free academic license for to use this particular applications. All right. So this is all about the GenDoc suite. So any questions till now with respect to this particular software? Are you able to understand this navigation, or you can may if you're not able to speak, you can may uh, add the same question on the chat itself as well. If
Yeah,
any questions,
if any
yeah,
questions are there, you can put in the chat box.
yes.
Yeah, sir. They have one actual inquiry mail ID related to this database.
Okay. So I've been shared an email ID in the same chat. Okay, so if there is no question, so we can going to be proceed for the next part of the demonstration. So this is the desktop based application that we have been demonstrated over here. Now I will also show it, and we are also having another version that is also completely free for the academics, where user can use, just a second, user may use the web applications of the uh, Jane suit application. So this is janedoc.geneforce.com. Sorry, I think
Sir,
so.
there are questions, I think. Dr. Ashwini Patil is asking that requested to provide the workflow for using application, including minute details like file format and all she's asking about.
File format. You mean the for the installation of the same file or the output that has been generated after the docking?
Sir, it is related to the input. Means which
In
kind of files are necessary to
Okay,
for the
okay,
input.
okay. All right. So, in the file format, if you see for the protein, there is an PDB format that is you needed to, upload from the upload protein wizard, or you can along with you are also having a direct option that you can enter the PDB ID over here, right? So this is the option for the protein. Then the second option that is for the ligand, you are having an multiple options like you for the upload option you can upload the mol file, you can upload the mol2 file, you can upload the SDF file, and you can also upload the PDB file, right? So mol file, SDF file, then you are also having in the PDB file, so and mol2 file. So these are the different type of option that is available for you to upload the input ligand file. And also there is an add smiley, so you user also, if there is no file for the upload, you are having a smiley strings of that particular compound, you can directly upload that particular smiley over here. You just have to paste the smiley compound over here, and then you can click on the add smiley, the same smiley it will be added to over here, right? And depending on the, if you're having a number of ligands, you need to upload those over here, and those all will be reflected into the ligand information, same that you can check and you can take for the further prepare ligand option. So these are the basic option that user has to input a file for to perform the molecular docking simulations. Understood? Any other questions, ma'am, we have?
Uh, no, sir. Actually, in extension, Patil madam has asked that she was interested to know about both the input file, means input as well as output files.
Yeah, and in the output, the all the output that has been generated into the .pdb file, in even in the complex, as I said, if you see the working directory, this all the pdb file and .pdbqt, this both has been generated over here. In the workspace, you can see the prepared ligand that is also into the pdbqt that has been generated for the ligand. And then also in the models, the each poses you are having an PDB .pdb file of the each ligand that has been generated after the docking, and also the same PDBQT file is also there in the folder directory where this molecular docking application that has simulation that has been performed after the docking. So it will generate all the required output extension file, which is uh, basically standard format that is needed and that has been generated after the molecular docking with the Autodoc Vina and Autodoc. So similarly, those all are the available over here. So those you can use it. Okay, all right. So next, that is the JNDoc suite. That is in another web server application that we also have. That if the user does not have the license for the desktop application, so this is a very best option for the people that user can just have to register free on this particular platform. They can register from this particular register button, and if the user has been already registered, they can directly log into this our tool. So I can just quickly log in for the demonstration. So see, this is my dashboard over here. It will reflect all the different type of docking that I have been performed. So let me quickly demonstrate this particular tools because we also have two other tools that we have to convert, right? Cover. So in the upload protein structure, you also have over here, there are the different options are available. First is the protein structure that you can upload the protein structure directly from the local directory. And then also if you're not having the structure of that particular PDB, then you can click on that particular PDB, download RCSB button, and in that particular it will ask you to enter the PDB ID, then click on the fetch protein button. So once you have been click on that, it will retrieve that particular protein directly from the web server. You can also when you click on this, you can select the same protein on the RCSB side. Once you have been retrieved, you can see that particular in the viewer itself on the web applications, right? There you can see the different chains, the outer molecules that are present, also the different ligand or the ions that are present in that, right? So you can click on continue with this protein, so it will save and continue with this particular protein. So it will in the next it will ask you to prepare your protein. So in this particular preparation option, if you see, there are the different chain, but for the docking you need an on only chain A and you wanted to delete the chain B, you can select that particular chain over here. Then you can click on the delete chain button and click on delete chain. So that chain has been deleted from over here, only chain A is reflecting, and the same has been loaded into the viewer itself, right? And there are the different preparation options that you can select, that is the add the hydrogen, the Coleman charges, remove the water molecules, remove the hate atoms, remove the non-standard residue, repair the missing atoms. So these are the basic preparation option that is available for the users that you can select over here. Then user can click on the prepare protein button. Once you click on the prepare protein button, so it will start to prepare that particular protein for the docking itself, right? So you need to wait for some time until the protein preparation will be completed. Once the protein preparation has been completed, it will ask you to upload the ligand. So see over here, there are the different options. You can directly upload, or you can draw that particular ligand or enter the smiley over here. So there are the different options. Under the upload option, you can see you can upload the .mol file, .mol2 file, .hdf file, and .pdb format, the supported PDB format that has been given over here. So any of this particular format you can directly upload. So for the demonstration purpose, I will select any of this particular ligand that is in .mol file. Then I will click on the upload ligand button. So see, the same ligand has been uploaded over here, right? And also if you're having a multiple ligand for the molecular docking, you can upload that particular ligand that will be reflected over here. There is also one of the option for the ligand preparation option. You can expand this particular options. Over here, there are the same option like the hydrogen, generate 3D coordinates, energy minimization. You are having an option to select the force field that you can select over here. And once you have been added the ligands in the this particular uh, window, then you can select the which ligand that you wanted to select for the perform proceed with it. You can select over here. If there are the multiple, you can select all button, so all the ligand where get selected which has been uploaded by the user itself. Then you can click on continue with grid settings. So once you click on that, the similar, if you see, you are able to see the grid box. So there are the different options for the grid selection uh, over here. One of the geometric center that has been selected. Then next is the crystallized ligand based and the manual selection option. For now, let me select the crystallized ligand base. Over here again, you have to enter the protein ID over here, then click on the fetch, right? So once you have been selected over here, it will fetch all the crystallized ligand. The similarly to the desktop application that we have been seen, right? It has like all the options for to all the options for to select the crystallized ligand where you wanted to perform the molecular docking within that particular receptor or the protein itself. So for now, we have been deleted at chain B already. We already have the chain A and chain B, and under the chain A and chain B, we can select this. So for chain A, let me select this particular ACP or the PETA. So you are having both the option. When that particular option that will be selected, it will automatically reflect that particular box, grid box within that particular uh, cavities directly, or the active site within that particular receptor. So let I have been selected ACP. So this is the grid, and also if you wanted to expand this particular grid box a little bit, you can click on grid control. So when you click on that, you can manually adjust this particular position over here, right? If you're able to see over here. So let me click on continue button. So once you click on that, you can give the name for this particular job, that is like any name that you can give. You can give the ligand name as well. Like I'm just for demonstration job one. You can select the number of exhaustiveness for the docking, number of binding modes, the energy mode. Then you are having the same option like which scoring function that you wanted to use for the molecular docking simulations. There is the Vinado and Autodoc 4. So as in fast, I can select the Vina over here, and once you have been selected, you can click on the start docking button. So the job has been submitted successfully. The docking simulation was running. So you need to wait for some time. The speed and the docking simulations, how much time it will take, it totally depends upon the receptor, the type of receptor that you have been selected, the length of receptor or the size of receptor that I mean, and the ligand that have been selected. If there are the multiple ligand, that usually takes some more time for the docking application. But once the docking application, sorry, molecular docking has been completed, you will receive on one email that your job has been completed successfully, and you can just go back and go to your dashboard and check the recently submitted job, and you can analyze this particular result. Now, if you see for over here, the the molecular docking has been completed for this particular receptor and the ligand that have been uploaded over here. Then I can click on the view result button, and once you click on that, see all the result has been generated over here, right? So if I able to see, so these are the interaction that has been shown over here, and you are able to see these are the -7.5. This is the highest binding affinity for the other rank pose that we can see over here, that has been loaded into the in this particular viewer. To understand the ligand, there are the different options over here. You can change the style, you can change the same into the full screen as well. You can hide the contacts, you can specify the different style for the protein and the ligand. So let me change the ligand color. Over here you can see the blue color has been added for the ligand, and all the interaction that are showing in this particular area. And also if you wanted to download this particular pose, so when you click on that, it will download the same pose. So .pdb file complex, that is the extension, the complex file of that particular pose will be downloaded. And also if you wanted to download all the complexes, then you can just click on this particular button. So all those particular result will be downloaded over here, along with the same zip file you also got on the email itself as well. If there are the multiple ligand has been uploaded, those will be reflected in this particular uh, window. So when you click on that, so under that particular ligand, there are the different poses that has been generated, or the binding mode that we can say, those will be reflected in this particular window. So this is how the very simple web application within the one click you can analyze and perform the molecular docking simulation in very easily. There is no the complex methods and that you need to. We also have the different documentations for the same. You can just for the new release which is recently launched version of the web application, so you can see how this will be work, which features that we have, which scoring function or the backend integration that we have been used, which supported file formats. You can see the detailed file format as the same question that has been arises. You can check our documentation. The same file format is also supported for the our desktop application as well. The protein, ligand, and output format. So these are the supported file format that currently available with us, right? So over here you can see the detailed documentations for this particular tool. Along with also we are having some blocks, like if you wanted to detail understanding for the different scoring function that is available for the GenDoc suite. that you can see over here. You can compare what how detailed comparison for the each scoring function, and the very important that you wanted to understand, like in which condition which scoring function that we should use. So there are the some blocks this has been given, at which process or the which step that you can select which scoring function. So you have to read this particular documentation in order to perform the molecular docking simulations. So this is all about the web application for the GenDoc suite. So coming to the very important part, that is our main website, that is the genforce.com. So once you visit this particular website, so this is the main page of our applications, sorry, our website GenForce. Over here we are having an more your tools under the, if you scroll down, we're having in Force ADME, that is for the prediction of ADME properties of your molecule. GenDoc, so that we have been already demonstrated. We are having a paraphrasing tool, that is the force base. We are also having an AI detector that would detect the uh, AI generated words in the manuscript or the any report that you have been uploaded. We also have a gene by predictor, that is the prediction of bioactivity for the molecules. We also have the gene mold, that in the first slide that I have been mentioned as in they know drug design. So this is the uh, basic version of the they know drug design of the gene mold, where you can just select the target, and based upon this given target, it will generate the different new molecules based upon the train data set that we have, or the algorithm that we have. We also have the Jane talks, that is the toxicity predictions. We also have the formulation AI, that is recently, I think, in the first session, Dr. Satish sir already taken this particular option. So you can all see over here. We are also having a detailed workflow for the Jane doc suite. For the this is 1.0 version, the latest is not uploaded, and different option that we can see over here. So let me first the quickly demonstrate on the ADME tool. So I'm just logging to my credential. If you are not registered over here, you need to register on this particular platform, and then only you are able to use this particular tool. Then I'm going to the Force ADME. In the Force ADME, there are the different options that you have to upload. You can either upload your molecule. You can directly click on the upload button and select the that particular molecule, or either you can draw your molecules over here, or you can add the smiley over here. So there are the different type of input option that you have, that you can choose among this particular option. So for now, I'm just taking one of our test molecule, and then once it has been uploaded, over here also you have in .mol, .sdf, and this ASMI, .smi, or you can directly enter the smiley. You can upload the multiple compounds as well. Once you have been uploaded, then click on Analyze uploaded files. So it will take some time, and once it has been completed, so it will show over here the batch analysis result over here. So this is the A1 molecule that I have been uploaded. Once you click on generate report, so it will the report has been generated. Or also you can, if you click on this view button, so this will, you can see the detailed analysis. So all the parameter like the log P, hydrogen bond donors, number of rotatable bonds, the polar surface area, this all has been shown. If you click on detailed report, detailed analysis, then you are able to see the detailed lipophilicity, the water solubility, the pharmacokinetics, the drug likeness profile for the drug, that particular compound, and some medicinal chemistry point that will be needed. So these all the parameters were calculated over here, and you are able to see over here. And if you see in the generated report, so the word report that have been downloaded over here, in the report, it will also contain the same data in more table in better view, that the same table that you can use uh, for the publications, right? If you see the table to which contain all the ADME properties with the table, and the detailed analysis with their interpretations will be added into the this particular report. So this is pretty, good report that has been generated and used by the different peoples that currently using this particular application. The next one that we have, the Gen Bio Predictor over here. Similarly, your user having an either they can for the bioactivity prediction, this particular tool. For this particular tool, we have been recently, that is version 2, which is based upon the machine learning model, which we have been used the random forest and XGBoost assemble model. We have trained almost 3,94,731 samples with the 43 targets that have been tested, and we have trained the extensive data set on this particular bioactive compounds for being taken. We are not only sit over here, we are also continuously updating the new version and increasing our database to train the model on the larger database, so it will generate the most accurate predictions and validations for the and predict the most reliable bioactivity for the molecules, not only for the bioactivity but the others models as well, right? So for the demonstration, over here you can either draw the molecules in this area, or you can enter the smiley, or you can upload the mole or the HDF file. So I will upload the same compound over here. Once you upload it, the same will be reflected into uh, the molecular editor as well, and the same compound name will be taken as a job name. Then you can click on the predict. So once you click on the predict button, thus you can see the bioavailability, the bioactivity score that has been generated, and then the target predictions. If you go over here, that there are the different target has been predicted. The anaplastic lymphoma kinase, EGFR, caspase-3, the mammalian target of rapamycin. These are the different. And in the property profile, if you are able to see the radar graph for the different property that has been, or the descriptor that has been calculated before to perform the actual prediction. So based upon this calculated predict descriptors, the activity predictions were completed. Then also the molecular property, that is the same like lipophilicity, hydrogen bond donors, acceptors, rotatable bonds, and the function sp3 hybridization that has been given over here. And also even the same compound was there in the public databases like the PubChem and Zinc. That link will be automatically pop up if the same compounds are available. For the novel compound, it will show as a no details over here, right? Otherwise, you can use this particular model, and this is very basic model that you can, very single click, you can also export the report into the different format that you wanted to export it. Right. So this is the Gene Bio Predictor. The next tool that we have, that is the Gene Tox. So that is the toxicity predictions. Over here also, user either can draw that particular molecule, or a user can also input the smiley strings of your compound that you wanted to perform the toxicity predictions, or you can upload the file format. So for now, I can just upload one of the ligand from here, and I can just click on the upload and analyze button. So see over here, the analysis within the second that has been performed, it will give you an different the molecular properties, as well as the acute toxicity, what is the LD50, what is the toxicity class of that particular compound, and the very important, that is the toxicity endpoint assessment, the active and inactive for molecules for that particular predicted toxicity. For the hepatotoxicity, this molecule is showing the toxicity, hepatotoxicity, right? It is showing the uh, active for the same. Also, the probability has been reflected over here. Also, the for sorry, carcinogenicity is showing as an inactive, right? So these are the different assessment for the different type of toxicity that has been shown over here, and active and inactive, and the probability will reflect that whether the compound is active for that particular toxicity or not. So these are the different options are available under the Gen Talks, or either you can also download that particular report when you just need to click on the download report button. The same will be report will be downloaded over here, and you are able to see the detailed report for the predictions of that particular molecule. This is one single table will be generated for the publications, and another that detailed analysis for this single compound. Even though if you upload the multiple compound, those all will be reflected over here, and the prediction and probability and reliability will be reflected over here for the each prediction. Those you can use and analyze for your uh, for to compare with the different types of the result, right? So the next that we have, that is the Jane mold that we are going to be cover. So this is the new tool that will generate target-based molecular generator. This is the one of the type of de novo drug design. Over here, first user has to, we are having some database of the currently available targets. So you can select, for example, I will select as an acetylcholinesterase, this Alzheimer target. You can just click on search. Once you have been select, this particular target has been available, right? Once you have been selected this particular, you have to select the different levels. So depending on this particular level that you have been select, the number of molecules that has been retrieved from the databases to that will be used same for the pre-trained model. So for example, I've been selected as a level 5, so the 5,000 molecules that will be retrieved for to generate the new molecules. So when I click on generate novel molecule, that job has been submitted successfully. And if I click on analyze history, so this will be in the queue job. That means this is running. You need to wait for some time until this particular uh, analysis were completed or not. So I will say show you some recent completed job application. This is I can just click on view result. So this is also one of the target that have been selected over here. If you see the different type of novel compound that has been generated over here. So this is our base version for the molecule generator. We are also along with moreover and adding in more options for you can add the selectivity and different fragments for the each generated molecules, and simultaneously you can perform the same molecular docking of that particular selected compounds we are in. That is in complete lab platform over here in virtually screen. You can virtually design and screen the molecule for the that particular selected target. This is the table view, all the generated molecule, their formula, their molecular weight, and other descriptor that has been calculated will be reflected. When you click on action button over here, you just have to can see the detail with the structure of that particular molecules over here. So these are the all basic things or the with respect to the Jane mole that you can perform. You can also you know um, explore other tools as well, but within the time constraint, that's why we have to cover up the basic demonstration for this all the tools. Now I'm going to be cover the very interesting tool that we just have recently live. We haven't yet this is completely in the beta testing right now, but this is also completely available freely. So to use this particular tool, this is somewhat Synthon doc application. This is the pharmaceutical crystal screening application. If you already registered for the this particular our application genforce.com, if you registered for this particular site, you can just go to synthondoc.genforce.com and you can use the same credentials for the login. There is no separate registrations were required for this particular tool. Once you click on login button, it will open this particular tool. Over here, this application is completely for to screen the selection of the crow crystals, right? This is one of the things from the pharmaceutical solubility enhancement. When you have to perform the solubility enhancement of any API or the drug, you have to be screen that particular compound based upon the different techniques. So molecular docking is one of the aspect that we will perform the docking of your drug molecule and the coformers. So these are the different additives, right, that is available. So we will dock this particular molecule in different ratios, and then we will going to be analyze how they are forming an interactions, how there are the number of hydrogen bonds, other interaction that will form when we are going to be use those particular molecule in different ratios, or the co-formers in the different ratios. So over here user can just upload their drug molecules. So this is erlotinib that have been downloaded from .hdf file, and this has been selected. Then we are having an 54 available type of different co-former. You can select the multiple type of the co-formers over here. For example, I have been selected as an urea, that is one of the co-former for the solubility enhancement. That when you have selected this will be reflect the which type of ratio that you wanted to select. So you can select 1:1, 1:2, 1:3, 2:1, and 1:4. So for now, I'm just select a 1:3 ratio of that particular area, where the one drug molecule and the three different ratios of the urea that has been taken. And just you need to click on the start docking button. So once docking has been started, it will prepare your drug molecule. Then it will prepare the different ratios of the selected co-formers. It will perform the sequential docking for the each particular selected that particular co-former, and then it will also generate the visualization state that in that particular visualization it will show you how this all the three ratios were going to be interact with the drug molecule. And depending on this is the first step for the screening, like you are having an more than like 20, 30, 40 co-former, and you wanted to select the one co-former that will be more, that will show the more the possibilities for the uh, forming an interactions and increase the solubility of that particular drug molecule, along with the practical implication that you can perform over here. So if you see that docking has been completed over here, and if you see the result has been generated, as I say, there are the 1:3 ratio have been selected over here. And if you see the minus 4.67, this is then binding energy that has been generated over here. And let me change the color for the co-former. If you're able to see, the blue color are reflect the urea molecule, and those where have been formed the interaction with this particular our ligand. So let me change the color for our ligand into the red color, right? So the red color are our erlotinib molecule, that is the BCS class 2 drug, and the others are the urea molecule. Those were formed interactions with this particular molecules when we have been selected 1:3 ratios over here. So this is a very early screening that you can use this particular tool for the application. And once you have been, you can, there is also export option. There is no, this particular result has been saved, you need to download this particular result in the zip file, or either you can export the word report. And this once you exported this particular result into the word, so you can see the this particular report. This will show all the details of the docking, what is the different docking poses that has been generated. Along with, you can find the which are the different bonds that has been formed the interaction, what is the bond type and category of that particular bond. So this is a very basic application that you needed for the screening of the co-former and the selection of the best co-former for the solubility enhancement. Along with, you can also add the label over here. In detail, you can analyze which type of the atoms has been interacting with each other, and those will be very easily you can interact with. You can just take the screenshot. There are the different option PND, HD, 4K. There are the different publications requirement that is available. And also, if you go forward, if we are having an references for that, in the detail we have been given for which algorithm that we have been used, which references that were taken while developing this, the applications with the supramolecular synthon approach for crystal screening, and those all the references were given over here for the development this particular. This is completely in the beta testing, but user can do use it according to your case of studies. As I think we already have crossed over time, so this is all about the demonstration that we have been cover. And I will also like to thanks all the management and the principal for inviting me here for this FDP program. So this is really a great honor for me, and thank you so much. And this is a really a based platform for everyone to be, understand, and you can use this particular application. And as I said earlier, this is an open platform for everyone. Whatever the tools that we have been demonstrated over here, you people may go and log in, register, and you can use it. You can share the feedback, and those the user who needed a desktop free application for the academic. So just send an email to our email ID that has been given, and we will going to be provide you the license for the same. Thank you so much. Over to you, Gayatri madam.
Um, yes, thank you so much, sir, for this valuable session. We have one question regarding
Mm-hmm.
docking, I think. Uh, Miss Fatima, she is asking that she is trying for plant chemical constituent docking, but she is getting value of docking score around about 4 and 5. Proteins are NAM and NAG, and the receptor is chemical constituent for means carvacol. So it's a drug, small drug molecule. So she is not able to understand what to do next.
So, in case of the phytoconstituent docking, the docking score is completely depending upon the selected ligand that you have been. If there is a very small molecule, so definitely the docking score will be very less. But along with, you have to understand in the selected protein what are the key amino residues, which is very crucial for to show the bioactivity. And also the very important point, that is the validations, that actually I missed to mention because of the time constraint. But see, you also have to perform the docking of the standard reference ligand. You need to redock that particular co-crystallized ligand that you have been selected, right? Once you have been redocked, you need to validate the RMSD values of both the ligand, whether the same pose has been generated or not. What is the RMSD? If the RMSD of both the ligand will be less than 2 angstrom, so definitely that is the best pose that you can select for the further docking. Along with, you need to understand what are the key amino residues where your the molecule that has been formed, the crystallized ligand has been formed, the interactions, and you need to analyze with your ligand of choice with whatever the same residues were form and interactions or not. So this is the key basic thing that you have to understand. And also, you if the score is very late, that need to modify, like you need to think like what is the orientation that you have to understand in the that particular compound that will maximize it, the interactions. So there are the part of the amino acid interaction that has been formed along with the standard compound. And the very important, you need to be prepare the different derivatives of that particular compound, so that will be form a more better interaction with the compounds. So I hope I answered your queries.
Yes, sir. And if we could share your contact number in the chat box, so rest people can ask their queries directly to you and contact you back regarding their further investigations. Sir, please share your mail ID and
So yes,
contact
yes, I
number
will
in the
share
chat box.
my
Yeah.
yes, yes, I will share my email ID over here. So people, those having any question, they can reach out to, they can send an email. So definitely, I will support you for the queries.
Yeah.
Yeah.
So moving towards the conclusion remark for this particular session, today we have the last session of EFDP and ESDP, which was enlightened by Mr. Ganesh Vaghure sir with thorough procedure which is needed for the docking, because this in silico studies are not only required only for the synthetic molecule, but now they are needed for phytoconstituent, as well as they are needed for even knowing drug interaction or drug sorry, ingredient interactions in that sense. So definitely, and as per my experience, we need to see for many of the tools to come up with the end result of the docking studies or in silico studies all over, I would say. So you need to club up number of tools together. So with this GenDoc or GenForce suite, what you have just discussed with us, sir, definitely will be helpful for those who are struggling with their results. And one genuine question from my side, actually, sir, what are the chances of accepting the results? Because many of the times it happens that if we use a freeware, journal are hesitating to or they are asking for the commercial software like that. So what your experience is about this?
Yeah, thank you for raising this particular question, because also I can see one of from the chat box there is a similar question that they have been asked. So the tools and the backend, the function that we have, scoring function that we have been used, that is actually already an extensively validated scoring function. And secondly, we are having an more than 4 to 5 publication on and Q1 and Q2 journal, especially in the Springer and elsewhere, where the same application that they have been used for the molecular docking and all. So definitely we will share those all the publication with you. So if you use our application in your study, you can cite those particular tools, sorry, that particular publications in your study. So there is chances of acceptance of your papers for the publications.
Yes, sir. Thank you so much. And with this, we have concluded today's session. And sir, it's a request to you to stay with us for the valedictorian session also that we are beginning just now with this